Ischemic and hypoxic injury occur because of diminished blood supply or decreased blood supply in particular tissue as injured tissue faster than hypoxia. Hypoxia first affects the cell's aerobic respiration in the mitochondria. Oxidative phosphorylation by mitochondria is affected because of diminished oxygen. Consequently, reduced oxygen tension reduces intracellular degeneration of ATP. So, decreased oxygen, hypoxia, affect aerobic respiration in the mitochondria, affects the degeneration of the ATP or depletion ATP. These have different effects in the system or in the cell. First, activity of plasma membrane. The ATP-driven sodium pump also is reduced because of reduced oxygen, hypoxia or ischemic. The plasma membrane affected and then affected the sodium-potassium pump, which reduced, then accumulation of intracellular sodium. The sodium will be more in the cell, and diffusion of potassium outside the cell. Potassium increase outside and sodium decrease inside the cell. This results in accumulation of water inside the cell, so this becomes cellular swelling. It's further complicated by accumulation of other metabolites such as lactic acid. Second, anaerobic glycolysis increase. This is the result of decreased ATP. So there is no ATP or decrease. We need another source of ATP. This from glycolysis. This leads to increase the AMP, adenosine monophosphate, instead of adenosine triphosphate, to maintain an energy of by ATP from glycogen from the liver, and thus depletion of glycogen stored. So when we use glycogen for ATP, the glycogen will be also depleted or decreased. Also, increase this glycolysis result in accumulation of lactic acid, and this lead to lower or decrease the pH. Also, decrease the pH and ATP cause detach of ribosome or infusolar ribosome, which is important in protein synthesis in the. This leads to reduction of protein synthesis. If hypoxia not relieved or not subside, not finished, the mitochondrial function and increase the membrane permeability, which lead to morphologic change like cellular swelling, that owing loss of osmotic regulation, and if oxygen restored, so will be no reduced oxygen, all disturbances will be reversible. If the cell reversibly injured, the restoration of blood flow can result in cell recovery. If oxygen or blood restored, or again will be good blood supply and good oxygen, enough oxygen, this leads the cell to be recovered and return to its normal function and structure. Means reversible. However, in some cases, still the tissue injured and sustain the loss of cells. In addition to this, irreversibly be damaged at the end of the cell. Sometimes this will be irreversible. This called ischemic reperfusion injury, which has clinically importance that significantly contribute to tissue damage in myocardial or cerebral infarction. So, ischemia and hypoxia will be irreversible injury when this clinically in myocardial, in the heart, tissue damage in myocardial or in the brain, cerebral infarction. The mechanism of this reperfusion injury, restoration of blood flow. The cell highly concentrated with calcium, swelling of the cell. Reperfusion in injured cells result in locally augmented recruitment of inflammatory cell or accumulation or increase of inflammatory cell white blood cells, which release high level of oxygen-derived oxygen species, free oxygen. This leads to membrane damage. Also, damage mitochondrial cause reduction of oxygen, therefore increase production of free radical species or free oxygen. Membrane damage, calcium concentration, and accumulation of inflammatory cell lead also to free radical, like also damage mitochondria, which lead also to free radical species. The free radical induced cell injury. The fluid is free radical, a chemical species with a single unpaired electron or electron hole attack the nucleic acid and membrane molecule تهاجم الممبرين الغشاء والنوم. Ischemic reperfusion, free radical induced injury, particularly that induced by activated oxygen species important in the mechanism of cell damage. This free radical damage also important in chemical and radiation injury, oxygen toxicity, cellular aging, mitochondrial killing by phagocytic cells, inflammatory cell damage, tumor destruction by macrophage. The reaction relevant to free radical induced cell injury. So when there is free radical, there is something happen. Lipid peroxidation, DNA fragmentation, affect the nucleus, affect the lipid, cross-linking of protein, also protein, ribosome, and chemical injury. Chemical injury induced by the following mechanism. Either chemical combined with molecular component or cellular organelle that lead to inhibition of ATPase that dependent transport and increase the membrane permeability like antibiotic or chemotherapeutic. So these chemicals affect the membrane permeability. Other chemical converted to toxic metabolites, which cause damage also of which membrane and cell injury direct by binding to protein and lipids. Reversible and irreversible cell injury within limit, as we said, the cell compensate the normal cell structure and function, and persistence of excessive cause the injury to pass to irreversible. This is associated with extensive damage to membrane, swelling of lysosome, mitochondria, and result in reduced capacity to regenerate the ATP. Extracellular calcium enters the cell, is stored and released, resulting in activation of enzymes and can catabolize membranes, proteins, ATP, and nucleic acid. Thus, and this, or hence, بالتالي, one of the earliest ultrastructural marker of irreversible cell injury is accumulation of amorphous, like calcium, in the mitochondrial matrix because of the affected ATP or membrane. After this is continued loss of protein, essential coenzyme and ribonucleic acid from plasma membrane depleting intracellular energy phosphatic energy to membrane, lead to their enzyme into cytoplasm, acid hydrolase to reduce pH of ischemic cell and degrade cytoplasmic and nuclear component. After cell death, so in irreversible injury, after cell death, cellular constituent or component digested by lysosomal hydrolases enzymatic الحالة of cellular and extracellular space. The dead cells may be replaced by large phospholipid mass called myelin figure, which phagocytes or phagocytosed by other cells or degraded into fatty acids and calcification of this fatty acid residue result in the generation or generation of calcium salt. So the cell death and then this cell death happen by digestion of lysosomal enzymes, and these dead cells replaced by phospholipid mass called myelin fibrils. And then again phagocyted by cells or inflammatory cells and will be changed or replaced to or degraded to fatty acids which calcified and into calcium salt. To summarize all these, irreversible cell injury events will affect oxidative phosphorylation in mitochondria and hence synthesis of what? ATP. Membrane damage is also a critical step in the development of lethal cell injury, and calcium is potentially mediator of final morphologic alteration in cell death. Irreversible affect phosphorylation in mitochondria and ATP and membrane damage. The mechanism of irreversible, there is two characterization of irreversibility. First, the inability to reverse mitochondrial dysfunction. عدم المقدرة لإرجاع الميتوكوندريا. And lack of oxidative phosphorylation and ATP generation. The ATP depletion is lethal even of morphological change. The development of disturbance in membrane function. The development in membrane function. Also, it is incredible the permeability to extracellular molecules and plasma membrane effect. So, important in irreversible, the ATP or mitochondrial dysfunction, also disturbances in membrane function. Causes of membrane damage, progression loss of phospholipid, membrane phospholipid, increase cytosolic calcium, which cause decrease in ATP, or cytoskeletal abnormalities, هيكل تغييرات غير طبيعية في هيكل الخلية، أو فقدان phospholipid membrane, which affect the ATP. Toxic oxygen radical, also lipid breakdown, membrane damage at the end result in massive leak of intracellular material and massive influx of calcium. Damage of membrane lead to تدفق الكالسيوم إلى داخل الخلية و swelling of the cell. Adaptation. We said under normal physiological adapted conditions, the cell adapt the change in the environment to physiological adaptation, which represents cell regulation by hormone or endogenous chemical mediator like enlargement of breast, and induction of lactation by pregnancy. Pathologic adaptation has some mechanisms and allows the cell to modulate their environment and escape injury. And this adaptation is stabilized between the normal unstressed cell and injured overstressed cells. Some adaptive responses involve up or down regulation of cell receptor. For example, those involved in the uptake of lipoprotein, and down regulation when the cells are cholesterol depleted. When the cholesterol is depleted, so the lipid is regulated by up or down regulation. More lipid or more cholesterol will be down regulated. Other adaptive response associated with the induction of new protein phases. The adaptive change in cell growth and differentiation. There is different of morphological appearance to adaptation. First, atrophy. The more it is shrinking in the size of the organ by loss of cell substance. The causes decrease the workload, immobilization to permit healing or fracture, حالات الكسور, fracture. Loss of innervation, like paralysis, diminished blood supply, نقصان or decreased oxygen, blood, inadequate nutrition, loss of endocrine stimulation, thyroid, aging. Hypertrophy is increase in size of cells and consequently increase in organ size. This is in contrast to hyperplasia, which is characterized by increase in cell number. In hypertrophy, the cell enlarged by increased synthesis of structural protein and organelles. It can be physiologic or pathologic. Physiologic, like uterus during pregnancy, increase the workload, like skeletal muscle or cardiac muscle enlargement, caused by increased functional demand or by specific hormone stimulation. Hypertrophy and hyperplasia can also occur together as obviously cause enlargement of organ as during pregnancy, the uterus, that because of estrogen stimulation of both muscle cells hypertrophy and smooth muscle cell hyperplasia. Cardiac muscle cells enlargement that occur with hypertension or aortic valve. This is example of pathologic hypertrophy. Hyperplasia means increase in size of organ due to increase the number of cells. It can be physiologic or pathologic. Physiologic, like hormonal hyperplasia, proliferation of glandular epithelium, like in female breasts during pregnancy. In compensatory hyperplasia, which occur when portion of tissue removed, like liver, or dissected. When partially dissected, the mitotic activity starts in the other remaining cells. It can also occur in the connective tissue during the healing and wound healing by growth factor stimulate the fibroblast blood vessel, also proliferation to facilitate the repair. Pathologic hyperplasia, excessive hormonal or growth factor stimulation after normal menstrual period, which cause increase the endometrial proliferation. And this regulated by stimulation of pituitary hormones and ovarian estrogen, and by inhibition through progesterone. If the balance between those or these two hormones disturb, endometrial hyperplasia shows common cause of abnormal menstrual bleeding. Increase the sensitivity to normal level of growth factors may also underlie the pathologic hyperplasia. Also, skin warts caused by increased expression of transcription factor by affecting papilloma virus. Hyperplasia remains controlled, and if hormonal or growth factor stimulation abates or finish, the hyperplasia disappears. The differentiation or this differentiated this process from cancer, in which the cell continue to grow despite absence of hormonal stimuli. After stop of the hormonal stimuli or the injury, the cell in cancer still continue to grow. Nevertheless, pathologic hyperplasia constitutes a fertile environment for cancerous growth formation arising. Metaplasia. It is a reversible change in which one adult cell type replaced by another adult cell type. The cell sensitive to stress, like epithelial metaplasia to squamous skin. In respiratory epithelium in habitual cigarette smokers. That normal ciliated columnar epithelium in the trachea and bronchi replaced by stratified squamous epithelium. This is epithelial hyperplasia. Metaplasia. There is intracellular accumulation. It is abnormal accumulation of substances in the cytoplasm or nucleus which cause different degrees of injury. This is also another morphological appearance of adaptation. We talk about abnormal or disturbances in the growth, and this is abnormal accumulation or intracellular accumulation. There are three pathways: either substance secrete normal or increase, but metabolically inadequate to remove it, like fatty kink in the liver, or normal or abnormal endogenous substance that genetic or acquired defect in its metabolism transport, abnormal exogenous substances like accumulation of like coal or silica. First, fatty kink, steatosis. It is abnormal accumulation of triglyceride or fat in the cells, in the cytoplasm of the cell. It is reversible injury. It can happen most in the liver, but also in the heart, skeletal muscle, and kidney. Causes: toxins, protein malnutrition, sun protein, diabetes mellitus, sukari, obesity, anorexia, and alcohol abuse. Significance of fatty kink depends on the cause and the severity. If it is severe, it will be irreversible or lead to cell death. This is a photo. You can see this is empty. This empty is full of lipid, but the lipid dissolve أو تذوب أثناء التحضير للشرائح، لذلك تظهر فارغة. Intracellular accumulation also cholesterol and cholesterol cellular cholesterol metabolism regulated by cell membrane, and overload with lipid become membrane-bound vacuole of lipid, like in atherosclerosis أو تصلب الشرايين in smooth muscle. Protein, it is less common than lipid, occur when the cell synthesizes excess amount of protein, like in nephrotic syndrome. Protein appear like pink, hyaline, droplet. It is reversible. Glycogen, also excessive deposition of glycogen inside the cell associated with abnormality metabolism in glucose and glycogen, and poorly controlled in diabetes mellitus or glycogen accumulation in renal tubular epithelium and the cardiac myocytes and beta cells of islet Langerhans. Also, glycogen accumulate in case of genetic disorder and glycogen storage disease. Pigments. Pigments, colored substance, may be exogenous or endogenous. Exogenous like the carbon from coal dust when inhaled or phagocytosed, like anthracosis that when the coal is inhaled and then phagocytes through macrophage transport by lymphatic to tracheobronchial lymph nodes and then aggregate. Endogenous include lipofuscin. This is brownish yellow, granular, intracellular materials, complexes of lipid and protein, accumulate of different tissue in heart, brain, liver. Melanin, it is endogenous brown-black pigment formed by melanocyte due to disturbances of tyrosinase enzyme found in the epidermis, البشرة, against the harm ultraviolet أشعة. Intracellular accumulation also hemosiderin is a hemoglobin-derived granular pigment مشتقة من الهيموغلوبين. It is golden yellow, brown, accumulate. This accumulation may be local or systemic excess of iron, زيادة أيون الحديد. And the iron stored in association of protein apoferrin called ferritin. The local excess of iron of hemosiderosis result from hemorrhage after lysis of erythrocyte. تتحلل erythrocyte. The red cells phagocytosed by macrophage and the hemoglobin catabolized by lysosome, and then accumulation of heme ion in hemosiderin. This is original red and then change to blue color of hemoglobin and then transformed to green blue by local formation of biliverdin and bilirubin. From heme, the ions or ion al-hadid of hemoglobin are accumulated are golden yellow. Systemic overload, then the hemosiderin deposit in different organ and tissues by condition called hemosiderosis. It is found in mononuclear phagocytes, liver, bone marrow, in spleen, lymph nodes, macrophage, in other organs, and parenchymal cells, but particularly in the liver, pancreas, heart, and endocrine organs. Hemosiderosis occur in the setting of increased absorption of dietary iron امتصاص الحديد or impaired utilization of iron or hemolytic anemia or transfusion. In systemic hemosiderosis, the iron pigment does not damage the parenchymal cell or impair the organ function. However, it will be extensive accumulation. It will be injured, including the liver fibrosis, تليف الكبد، أو في حالات الهارت فيلير قصور القلب، أو في حالات السكري diabetes. This is the hemosiderosis. Brown pigments. Also pathologic calcification. This is common process in a wide variety of disease. This is abnormal deposition of the calcium salts. It is two types, either dystrophic calcification or metastatic calcification. Dystrophic calcification, it is accumulation of calcium in dead tissue or necrotic tissue, like atherosclerosis, تسرب الشرايين. Accumulation of lipids cause organ dysfunction, and then for damaged heart valve or the aging, and then calcification. Pathogenesis of this involve initiation of propagation of both extracellular or intracellular. Metastatic calcification, this is occur in normal tissue whenever there is hypercalcemia. So there is hypercalcemia, increase in the calcium, so it is transported or moved through the blood to another part. There is 4 cases of hypercalcemia, either increased secretion of parathyroid hormone, destruction of bone, vitamin D related disorders, or renal failure in which phosphate retention lead to secondary hyperparathyroidism. This is aorta or aortic valve, and this white is calcium. This is under the microscope, so this is the blue color is the calcium. The morphology of reversible cell injury. The plasma membrane alternation, تغييرات بتحصل في البلازما membrane, bleeding, losing of intracellular detachment, mitochondrial damage, change like swelling, dilatation of endoplasmic reticulum or detachment of ribosome, تباعد أو انفصال ribosomes, swelling للخلية or mitochondria, bleeding, انكماش, nuclear alteration. 2 patterns of morphological change recognized under the right microscope. Cellular swelling, first as hydropic degeneration, water, hydro means water. Vacuolar degeneration, vacuoles, فراغات, small clear vacuoles, فجوات, small clear in the cytoplasm. This is reversible. Another sphatic change, this occur in hypoxic injury, the toxic or metabolic injury, lipid vacuoles in the cytoplasm, like in hepatocyte, the liver cell, or myocardial cells, the heart cells. Also reversible. So 2 reversible change or morphological change in reversible injury, cellular swelling and fatty change. This is cellular swelling. Here the cell is swollen. This is also irreversible cell injury. First, necrosis. Necrosis, it is morphological change follow cell death in living tissue. تغييرات ظاهرية تتبع لموت الخلية في الأنسجة الحية. Characterized by change in nucleus and cytoplasm. تتميز بتغيرات في النمط والسيتوبلازم. The morphological appearance has 2 processes, either enzymatic digestion of the cell or denaturation of protein. The hydrolytic enzyme may derive from cells themselves, autolysis, or from lysosome, This is heterolysis. The dead cells increase eosinophilia, will be more red and glassy homogeneous appear due to loss of glycogen, and the cytoplasm will be vacuum. So the change in the cytoplasm will be more eosinophilic, more red, glass or homogeneous. There is vacuum. In the nuclear, there is breakdown of DNA, pyknosis, تغلظ للنواة, shrinkage, انكماش, and increase the basophilia of DNA, and karyohexis, تكسر النواة, and pyknosis, تقلص النواة, and karyolysis, تحلل النواة. The nucleus in dead cells sometimes completely disappear. تختفي. Types of necrosis, there is coagulative necrosis. This means preservation of general architecture of the tissue. The cytoplasm of necrosed cells become more eosinophilic, more red, and nucleated. This is example as myocardial infarction. The liquefactive necrosis characterized of focal bacterial or fungal infections like abscess, and there is accumulation of white blood cells and then undergo lysis. Another is gaseous necrosis. This is encountered in foci of tuberculosis infection. The center is cheesy, like cheese, either in bacterial toxin due to bacterial toxin, primary or secondary due to ischemia and infection. There is amorphous granular debris appear like amorphous granular debris, حطام لخلايا عديمة الشكل with a ring, حلقة في التهاب حبيبومي or granulomatous inflammation. The tissue architecture is completely unclear. Fat necrosis, there is focal area of fat destruction, typically occur in pancreatic injury, like in release pancreatic enzyme, like in acute pancreatitis. Enzyme hydrolyzing triglyceride and then release the fatty acid, which combine with calcium salt, lead to cause white area fat saponification. So the fatty acid or triglyceride combined with iron. Histologically or under the microscope appear as shadowy outline of necrotic cells with basophilic calcium deposition by inflammatory reaction. In a living patient, most necrotic cells and their debris disappear by combined process of extracellular enzyme digestion and leukocyte phagocytosis. Phagocytosis by cells or leukocyte or white blood cells. The programmed cell death, another death of cells by programmed cell death or apoptosis is programmed cell death that occur in multicellular organisms. Several physiological processes include programmed destruction of cells during embryogenesis, queen again, or hormone-dependent physiologic involution, like in endometrium during menstrual cycle, cell deletion in proliferation population in intestinal crypt epithelium, the intestine, deletion of autorecessive in cell thymus, or a variety of mild injurious stimuli like heat radiation. The failure of cell to undergo physiologic apoptosis may result to unaberrant development or unimpeded human glorification. The morphology of apoptosis involves single cell or cluster of cells appear eosinophilic cytoplasm, chromatin condensation also. There is chromatin condensation. There is karyoxysis also تكسر النواة. And after this karyoxysis, the cell rapidly shrunk in the cytoplasmic buds and fragment into apoptotic bodies in vesicle. So this is the difference. After the nucleus is karyoxysis, after تكسر النواة, the cell shrink in buds or vesicle called apoptotic body. In contrast to necrosis, which is a form of traumatic cell death that results from acute cellular injury, but apoptosis generally confers advantage during organism's life cycle. مش مفيدة. For example, differentiation of fingers and toes in a developing human embryo, because the cell between the finger apoptose, and the result is that the digits are separate. So unlike necrosis, apoptosis produce cell fragments called apoptotic bodies that phagocytic cells are able to engulf and quickly remove before the contents of the cell can spill out onto surrounding cell and cause damage. The cellular aging. There is number of cellular function decline or be more reduced progressively with age, as mitochondrial oxidative phosphorylation, synthesis of structural enzymatic, irregular nuclei, accumulation. There is intrinsic molecular clock of cellular aging theory and extrinsic stressor of cellular environment, either extrinsic stressor or intrinsic molecular clock. The intrinsic cellular aging theory, there is two mechanisms. Incomplete replication of chromosome ends. So the gene would lose after a number of divisions. Another is clock gene, genetic timer control. The gene alters the growth rate and timing the development process. So the cell lifespan regulated by balance of ongoing injury, the ability of the cell to repair the damage. It involves the free radical, which reduce the mitochondrial and DNA damage and post-translation and extracellular protein. So there is intrinsic or extrinsic. Intrinsic, there is two mechanisms: incomplete replication, chromosome, the end or loss of chromosome after division, or clock gene or genetic timer control, timing the development process. In summary, the cellular aging mechanism involve both programmed event and consequences of progressive environmental injury. The programmed aging assumes predetermined sequence of events, including depression of specific genetic programs leading to senescence أو الشيخوخة.